Hi! Welcome to the Cherokee Beekeepers Club video regarding quality queens. In this video, you'll learn about the different factors that affect queen quality. A colony can't survive or produce excess honey without being queen right with a quality queen. It's important for successful beekeepers to be able to analyze the quality of their queens. So let's dive in. We're going to go over a few factors that affect queen quality. Those factors include the queen age and vigor, genetics, drone quality and mating conditions, pesticides, habitat fragmentation and climate change, pathogens, nosema, mites, and other pests, nutrition, and finally beekeeping practices. Let's take a closer look at the first factor, queen age and vigor. A queen takes about 16 days to develop from egg to emergence. Upon emergence, she spends 5 to 8 days in the colony maturing and strengthening her flight muscles. Newly emerged queens don't have the familiar fully extended abdomen of a mature mated queen. Once ready, the virgin queen will go on a mating flight about 1 mile from the colony. And during this mating flight, she will mate with 6 to 30 or more drones, with the average being about 12 to 15 drones. Inside of her abdomen, a queen has a sac called a spermatheca. After each mating, the queen will only retain about 3% of the sperm from each drone she mates with, for a total of about 3 to 5 million sperm when she's finished. This photograph shows you that a virgin queen spermatheca is clear because it contains no sperm, while a mated queen spermatheca is milky white from being full of sperm. The tracheal net normally covers the spermatheca and provides oxygenation to the stored sperm. However, it's been removed here so you can see the spermathecas more clearly. 3 factors affect the sperm quality in the spermatheca. First, the queen's age. A newly mated queen has 3.5 million sperm. After a year, she only has about 1.25 million. And at 2 years of age, she only has about 0.8 million sperm left. This huge drop in sperm count negatively affects queen laying and the ability of the colony to stay strong in numbers. A second factor is sperm motility. Now, in a newly mated queen, the sperm have tightly coiled tails and they spin super fast in circles inside the spermatheca. Over time, as sperm is used in fertilization, the sperm left behind in the sac have more room to swim around. So they uncoil their tails, but also their swim speed significantly decreases. Now this can affect fertilization rates and the strength of the colony. And then finally, premature sperm death can affect sperm quality. A queen may be poorly mated and not have a lot of sperm to begin with, but also a queen may have a lot of sperm, but if the drones with which she mated didn't produce high quality sperm, or if there's a problem with the queen's nutrition or with her tracheal net, premature sperm death can occur. If the sperm die, then the queen will become a drone-laying queen, which means she can no longer fertilize her eggs and can only lay drones. Without female workers being replenished, the colony will quickly die. Let's look at the queen's vigor. The queen has what's called the queen mandibular pheromone, QMP. It's distributed throughout the colony. A queen-right colony operates efficiently because of the QMP. A young queen that has a strong QMP has a large retinue, a group of bees who groom and feed her when she's standing still. This large retinue then passes around the hormone all throughout the hive when they travel, when workers groom each other, and through trophallaxis. That's where bees feed each other mouth to mouth. The higher the concentration of the queen mandibular pheromone in a colony, the stronger the colony. If a poor quality queen has less than ideal amount of queen mandibular pheromone, then she'll have a smaller retinue, and a smaller amount of QMP is distributed throughout the colony. Smaller amounts of queen mandibular pheromone weakens the colony. So if the QMP diminishes enough, you can end up with a colony of laying workers. Workers will also begin rearing queens and supersede your cells, and the colony will have lower productivity overall. Beekeepers should keep track of your queen's age. Mark them, Clip the queen's wings so you know how old she is and keep records. Also, replace your queens at least every 2 years with queens from a reliable source that you know will have the best chance of being quality queens. Let's look at genetics and how it relates to quality queens. Good quality queens come from healthy, high-quality queens that are well mated with genetically diverse drones. Going deeper, we note that a good, healthy queen should also have been bred for hygienic traits. A hygienic queen will produce hygienic offspring, who can reduce stressors in the hive, which results in a stronger, healthier colony. If a queen experiences inbreeding, this causes her to produce diploid drones. Diploid drones are blondish colored and they have very light eyes. They're not viable and their sisters will eat them. If they're not eaten, then they end up dying prematurely. And too many diploid drones causes a shotgun brood pattern. So if you see a shotgun brood pattern and you can't find a cause based on disease, you may have a highly inbred colony. Inbred colonies are weaker than genetically diverse colonies, so ensure your queen is well mated. What about the drones? Healthy colonies produce thousands of drones if the colony is large enough and if environmental conditions are right. Drone emergence and queen mating flights coincide. Drones form all of their sperm prior to emerging, but upon merging they have to wait about 12 days before the sperm migrates to the seminal vesicles. At this point, he's sexually mature and has about 10 million spermatozoa. Now, he'll go on multiple mating flights a day. He'll frequently come back to the hive to refuel and go back out again to what's called drone congregation areas, or DCAs. That's where he waits with other drones for virgin queens to fly by on their mating flights. When he spots a virgin queen and is able to mate with her, he will invert his phallus and his endophallus stays inside the queen. That's known as the mating sign. Now, every drone provides less than 1 million spermatozoa to a spermatheca. Now, besides diploid drones from inbred colonies, what else can affect a drone's ability to mate well with a virgin queen? Pesticides. Studies have found that sperm viability dramatically drops in drones raised on frames that were lightly coated with miticides and agricultural pesticides. For example, on frames that were coated with fluvalinate and coumaphos, the sperm was only 30% viable. That means that out of all the sperm in the drone, only 30% of it was alive, as opposed to control group drones, which had 100% viable sperm. With amitraz, viability was at 80%. With chlorothalonil and chlorpyrifos, viability was at 40%. Mating conditions can affect queen quality. If there aren't enough sexually mature drones where she goes to mate, then the queen will be undermated. and will not have a spermatheca full of diverse sperm. She will run out of sperm, and that will cause her to prematurely fail at laying eggs. So what can beekeepers do? Well, keep your drones healthy by changing out your frames every 2 years to reduce contaminants. Ensure that you aren't causing inbreeding. Be knowledgeable about the apiaries around you. So if you're rearing queens, then you're aware if there are enough drones in the area for mating. And be aware that if you live in an area where Africanized feral bee colonies reside, if you allow your virgin queens to go out and mate, then the sperm she acquires is likely to be Africanized. We have seen how pesticides can affect drones, but can they also affect queens? Studies on pesticides and queen rearing found a dramatic drop in eggs laid by queens raised in pesticide-laden wax. And let's also look at habitat fragmentation. Now, urbanization, or the building of housing developments and commercial districts, can wipe out a lot of native wild plant life. This creates food deserts, which reduce the quantity and quality of food availability. Now, climate change has a powerful impact on the behavior of plants and flowers. Climate change has been shown to cause early flowering and change the distribution of entire floral species. Changes in weather patterns can affect pollen and nectar availability, and also the quantity and quality of pollen and nectar. A change in food availability can weaken a colony's immune system, or even cause death by starvation. Finally, an interesting temperature study showed that during the transit of packaged bees, the packages experience a variable range of temperatures not normally experienced by a colony. Now, this fluctuation in temperature can negatively affect the quality of the queen. To help avoid this problem, try to chain your bee packages locally.
Every year, farms and fields play host to a symphony of sorts. Pollination, the springtime syncopation of flowering plants and the animals who feast on the nectar and pollen they produce. Over millennia, pollinators like honeybees have evolved a well-timed dance with plants. But now, plants may be changing their tune. Spring green-up, when plants wake from winter and sprout leaves. It's such a global phenomenon that NASA satellites can see it from space. Sensors, such as MODIS on NASA's Aqua and Terra satellites, can show us how green our planet is throughout the year. And they've captured something strange. In the northern U.S., spring green-up is starting about a half day earlier each year. The likely cause? Our warming climate. But is pollination also moving earlier? The images can't detect individual flowers, so scientists have been left to guess, until now. NASA research scientist Wayne Isaias spearheads a special team gathering data directly in the field. They're the honeybees in his Maryland backyard.
Honeybees are great data collectors for understanding processes of pollination. Bees fly 2.5 miles in all directions to scout for bee forage and bring back pollen and nectar. And so therefore they sample a very large range of environments.
By weighing the hives, Wayne can detect when nectar peaks and ebbs each year.
During the winter, the hive loses weight as they eat the honey to feed their babies and keep warm. And then when plants start blooming in abundance, the hive starts gaining weight. It can gain a tremendous amount of weight. I've had a hive gain 25 pounds in one day.
Wayne's been keeping tabs on his bees for less than 20 years, but in that time, pollination has moved more than 10 days earlier.
That's completely in sync with what the satellite data record shows, the world here getting greener earlier in the spring by about a half a day a year. If we have a few scale hive measurements with the wall-to-wall coverage of the satellite, we can then extrapolate the scale hive measurements of when the nectar flows occur to very large areas of the country.
Now, to get a bee's eye perspective of how pollination is changing in very different environments, say deserts or mountains, Wayne's doing a little networking.
HoneyBeeNet is a network of citizen scientist beekeepers that volunteered to weigh their hives to give us more data points, to see how the nectar flows are changing in all different parts of the country.
If pollination dates keep creeping forward, plants and pollinators could move out of sync. Currently, young bees are able to grow and get out on the hunt by the time plants bloom. But if plants bloom before bees are ready, both miss out. The plants don't get pollinated and the bees go hungry. But more than just bees might miss a meal. NASA satellites can help us understand how climate change might affect what's on our dinner table.
Modern agriculture requires bees as part of the production. It's as mandatory for food production as is pieces of irrigation pipe and fuel for tractors. So if we're to understand the impact of climate change on our ecosystems, we must understand how this plant-pollinator interaction is being impacted by climate change.
Let's look at how pathogens and pests can affect queens and queen quality. Now, Varroa destructor feeds on fat bodies and hemolymph, and a vector of several bee viruses that are transmitted to drones, workers, and queens. Queens can get the deformed wing virus. They also can get the black queen cell virus, where affected queens don't emerge, they're blackish, and they're removed by workers. Now, Nosema cerana is a gut pathogen that creates digestive issues like dysentery, and it can affect queens. There's more stress in a colony due to Nosema infection, and the quality of the queen mandibular pheromone changes significantly in queens affected with Nosema cerana. It makes the queen smell differently, and the workers are less attracted to the queen, which causes a significant drop in the distribution of the queen mandibular pheromone in the colony. Queen nutrition is important and affects the quality of the queen and the colony as a whole. Humans can't live off hamburgers. We need diversity in our diet, and the same goes for honeybees. Consuming the same pollen source is bad for bees. They need pollen diversity to be healthy. Now, a study in 2010 found that dietary protein diversity, known as polyfloral pollen, has a direct impact on immunocompetence. Polyfloral diets boost colony immunity, and queens who consume diverse natural pollen have been found to lay more eggs than queens who have consumed pollen patties. Queens are also found to have a larger queen mandibular gland if fed diverse pollen rather than just pollen patties. And beekeepers should plant pollinator seed mixes to introduce a variety of pollen to their honeybees, and then save pollen patties as a booster rather than a regular protein source for your colonies. Let's take a look at our beekeeping practices. If we don't keep track of our queens and care for them properly, we're inviting swarming, supersedure, drone-laying queens, and laying workers. Ultimately, by ignoring our queens, we ignore our colonies, and ignored colonies will weaken and die. Laying worker drones are inferior to drones from quality queens. The traits of laying worker drones are greatly inferior to queen drone brood. If we have a laying worker colony and ignore it, then we're introducing tons of inferior drones into the environment, which will end up mating with queens from your neighboring beekeepers. So don't ignore your colonies or their problems, because that affects everyone around you. Now, all the factors in this presentation are reasons why premature queen supersedure in the U.S. is relatively common. Supersedure is what happens when a colony decides to replace the reigning queen with a new queen. They do this when they sense that the current queen is no longer a quality queen. So to help keep your colonies healthy, requeen every 1 to 2 years. You'll end up with a higher colony population and more honey. Also, your colonies will be less likely to swarm. Thank you. We hope you've enjoyed our presentation on queen quality. You can dive deeper into the subject of queen quality by searching for published online studies that are available on the web. Use the search terms honeybee queen study. Thank you and happy beekeeping.